Hello everybody, my name is Amir and this is Learnoplasty, in which we make your previous learning beautiful and understandable with a surgery. This video is about alpha helix. Alpha helix and beta sheets are called regular secondary structures because they are composed of sequences of residues with repeating phi and psi values, which we discussed in our previous video. So before we begin, make sure you have watched our previous video about peptide bonds and those torsional angles. So here we have an alpha helix. First of all, notice that the R groups of the backbone amino acids project outward and downward from the helix in order to avoid steric interferences with the backbone and the R groups themselves. Then you should see that each peptide bond between two residues rises 1.8 angstrom and rotates 100 degrees, so that means 3.6 amino acid residues exist in a turn. Therefore, you can realize that the amino acid residues spaced 3 or 4 apart in the sequence are spatially close, quite close to each other, while those spaced 2 apart are actually situated on opposite side of helix. For instance, take a look at this left picture. This is the view you would see if you look down the longitudinal axis. As you notice, the residue number 1 is quite close to residue number 4, so they should be compatible with each other. I mean, in the case of polarity, for example, at the pH of 7, these amino acids cannot be both glutamic acid because at this pH they are glutamate and have a negative charge that actually tend to repulse them, so they cannot be this close. Or one cannot be a polar amino acid and the other one hydrophobic. So the nth residue and n plus 4th or n plus 3rd residue should be compatible with each other in case of polarity. But if you take a look at the second residue and the 4th one, you'll see they're on the opposite side. And this is not true just for the R groups. It also makes sense for the backbone too. In fact, the oxygen in the carbonyl of residue N bonds with the hydrogen of the amide of N plus 4th residue, and that is the hydrogen bond that stabilizes the alpha helix. Alright, now let's talk about the screw sense of the helix. By screw sense, I mean the direction that a helix turns around its axis. There are 2 ways to know the direction. Number 1, to look down the axis and see if it is clockwise, which is right-handed, or counterclockwise, which is left-handed. The second way to understand is to put your thumb in the direction that the helix rises and make fists of your 2 hands, and then the direction in which the other 4 fingers are curved is the screw sense of the helix. You might have already guessed that amino acids act differently in the helix as they have different R groups. For example, branching at the beta carbon atoms, as in valine, threonine, and isoleucine, tends to destabilize a helix because of steric clashes. Or aspartate, asparagine, and serine have hydrogen bond donors or acceptors in their R groups close enough to their main chain so that it may compete with carbonyl or amide of the main chain for the hydrogen bond and disrupt the helix. Because of its structure, alanine shows the greatest tendency to form helices in most experimental model systems. So this table shows the difference of free energy of an amino acid residue to participate in alpha helix relative to alanine. And as you can see, proline, because of its cyclic structure which lacks NH group, is actually a helix breaker. Or glycine, because it has more conformational flexibility due to its small R group, rarely participate in alpha helices. And that is that. Don't forget to subscribe to the channel. Thanks for watching. Bye bye.